My name is Amy Sullivan, and I'm the current president of AAAR. And it's my pleasure to welcome you to the 35th in our monthly AS&T lecture series. This is a new initiative for AAAR being supported by the Freelander Memorial Fund. So with these lectures, we hope to highlight the amazing research happening in our community, tie our journal to other activities, and also give us an opportunity to meet outside of the annual conference. So each month, the editors of AS&T select a high-impact paper to be presented by its authors. In addition, each of these lectures is being hosted by one of our AAAR student chapters. With that, I want to thank everyone who's helped to make these possible and all of you for joining us. Now I'll pass it off to our student chapter from the University of Maryland, College Park to get us started. Thanks.
Thank you, Amy. My name is Esther Olonimoyo, and I'm the president of the AAAR UMD student chapter. And I'll be introducing our guest speaker for today in person of Dr. Zhehong Tian. Zhehong is an early career researcher having recently completed a PhD in aerosol science with the EPSRC Center for Doctoral Training in Aerosol Science at the University of Bristol. She's now based at the University of Hertfordshire as a postdoctoral researcher fellow with a research focus on real-time detection and characterization of bioaerosols using light-induced fluorescence technique. And with that, I'm going to pass it on to Zhehong. And a quick reminder is that if you have any questions you'd like to ask, we have time at the end of Zhehong's presentation to take your questions and also have some interesting discussion. So hand it over to Zhehong.
Thank you, Esther. Let me just share my screen, and if you can tell me you can see it okay, would be great.
Yes, we can
see my screen.
Yes.
And it's in the presentation mode. Nice. Yeah. Okay, so hi everyone, I'm Jianghan. Thank you very much for having me today, and I feel really excited because all of a sudden I've seen a few familiar names in the participants, and so as Esther introduced, I did my PhD with the EPSRC Center for Doctoral Training in Aerosol Science between October 2020 to 2024, and early this year in January I earned my doctorate. Now as a postdoc research fellow, I work in the University of Hertfordshire, so this is my new address. And if you never heard of it or you don't know where it is, it's basically north to London, and it's about 25 minutes direct train to central London, actually. This is where I am now, and I would like to present my paper on the AS&T titled The Microphysics of Surrogates of Exhaled Aerosols from the Upper Respiratory Tract. And I would like to also mention all my collaborators, including Rob Alexander, Dan Hardy, Tom Hilditch, Henry Osborne, Alan Hadrow, and my supervisor, Professor Jonathan Reed. So without them, I couldn't have done all of this work. And as you can tell from the title, I will be talking about the micro physical measurements of surrogate respiratory aerosols instead of real human aerosols. And also the aerosols will focus on the upper respiratory tract instead of lower respiratory tract, such as lung fluid. Okay, so as we all know, airborne disease transmission and respiratory droplets, they can carry infectious pathogens and transmit respiratory diseases such as COVID-19, and we have witnessed the importance of understanding airborne transmission around 2020. And typically we have an infected person that generates aerosols that containing infectious pathogens, and depending on the size of the droplet, if it's bigger, it might, fall onto the ground. And if it's smaller, it can be suspended in the air. And through the, transportation process, it can shrink into much smaller sizes and can be inhaled by another potential host, for example. And my talk my research, my work here is focusing on this particular process, which is the exhalation process. So exhalation is actually a highly dynamic process. So in our human lung, due to the different conditions in the gas phase conditions between our lung and also the ambient air. So typically here I listed the differences between lung and ambient conditions. And in our lung, we have a extremely high CO2 level, which is about 5% atmosphere or 50,000 ppmv of CO2. However, in the air, typically we have a 400, or in a crowded space, it can go up to, a few thousand ppmv CO2 level in indoor settings. And in our lung, it's really humid, so it's 99.5% RH relative humidity. And in the ambient environment, typically we have maybe 40 to 80, and depending on where you are, for example, in an office space setting, it might, controlled to a 40, 50% RH. And during exhalation, so because the droplets are experiencing such different environment, so we can observe, for example, the water evaporation, and this might lead to, depending on the concentration, the composition of the droplet, it might cause the phase change, and also because of the CO2 concentration is really high, actually it's partitioned as bicarbonate in our respiratory fluid. And when we exhale these CO2 in the liquid, so aqueous CO2 has a tendency to evaporate during the exhalation process, and this will cause the pH rise in the respiratory aerosols. And depending on the ambient conditions, depending on how much acidic species are there, the pH of the droplet might drop again. So here is just to show the physical properties of respiratory aerosols. This is I took from a study that measures the exhalation particle size distributions from 132 subjects, and they were doing different respiratory activities. As you can see, different depending on where the particle is generated and what kind of mechanism it is. We typically have different size ranges of these droplets. For example, breathing is typically from lower respiratory tract. They have some much smaller sizes. And if we are doing, for example, vocalizing a process like singing, speaking, shouting, these are typically, generated from the oral cavity, laryngeal, and they has tens of micron in size. And this is the size range I will be talking about, the size regime I would be talking about today in my measurements. And also here, this is study that shows the composition. So not only the size distributions are different, and also the compositions also varies between, for example, saliva and lung fluid in here. So here you can see there listed a list of ions that existing in saliva and lung fluid. We can see the distribution is quite different, and this is based on mass concentration. You can see here the black shade is protein, and however, in lung fluid there are a lot more proteins compared to saliva because the surfactant, and also this orange color indicates bicarbonate, and the green is sodium, and all of these, they are quite different. And here they also showed the collection media. So typically, if we want to grow virus, we need to put them in a collection media or suspended media in order to cultivate them. So in this slide, I would like to show the So in my actual work, I measured artificial saliva recipe from Wu et al. Study, and also Dulbecco's modified Eagles medium, which DMEM, which is a type of cell culture media that are widely used for cultivating SARS-CoV-2 virus. And how do they compare with the real respiratory fluid, for example, saliva, nasal fluid? And here we can see that in terms of the inorganic salt, it shares quite a similar compositions. For example, potassium chloride, sodium chloride, calcium chloride, and sodium bicarbonate. The salt. And in terms of the organics in respiratory fluid, we have mucus, and in a smaller, it consists of mucin, and then we have different enzymes. But in these recipes, they don't really have this. But in artificial saliva, we have the mucin and a very tiny amount of DMEM existing. And in the Dulbecco's modified DMEM solution, we have glucose, amino acids as the major organics. And comparing the pH range of these solutions, so you can see the respiratory fluid is, so I got this data from a paper between 6.2 to 7.6. And then artificial saliva has a pH 7, very neutral pH, and then DMEM has a slightly alkaline, 7.2, 7.4, but they are all covered by this pH range. And in terms of the pH buffer systems, so how does the pH regulated in these solutions? So in real respiratory fluid, we have bicarbonate as the major buffer, and then phosphate as secondary, and protein as the other buffers. And in artificial saliva, we have this bicarbonate and phosphate existing. And also in DMEM, we mostly have the bicarbonate to regulate the pH of the solution. So this slide demonstrates the effect of evaporation on surrogate respiratory aerosols. Sorry, to start with, here I listed, the components in the respiratory fluid from the literature and what their concentrations in gram per liter. So these gray bars is a summarization of, the variations from different participants. I believe this is probably from 50 in total. So from these 3 studies, so they have taken data from 50 participants, and they measure the calcium, sodium, potassium, chloride, all of these ion concentrations, and you can see it's quite varied. So yes, it is varied across individuals, and even for one individual, depending on our activities, depending on whether our saliva is activated, we generate different amount of ions. For example, when we're eating, we generate a lot more compared to when we are resting. And on the right-hand side, these are the solutions I measure or will talk about in the later slides, including the most basic salt, sodium chloride, and artificial saliva formulation from Wu et al., and the minimal essentials medium, which is also a cell culture media similar to DMEM, that are used to cultivate virus. And these bars are, these lines are just illustrate which level do they at in terms of these ions. You can see they are quite varied. So the way to study the droplet dynamics, or the microphysical measurements, is by using a technique called the Comparative Kinetics Electrodynamic Balance Technique. So this is or CKEDB. So this is a well-established technique based at the University of Bristol. So typically we have 2 micro dispensers or droplet on-demand dispensers to generate single particles. So we use orifice diameter about 30 micrometers. So the droplet we generate, the initial size is typically around 30 micrometer, or between 25 to 30, I would say. And then the droplet is generated, so a charged droplet will be trapped in the electrodynamic trap, and we use a 532 nanometer laser to illuminate these particles, and we collect the elastic scattering at a 45-degree angle, which is, compared to the forward scattering. And then within this chamber, the humidity is controlled by combined, wet and dry nitrogen gas, and then also the typically we can control the humidity between 0 to 99%, and also the temperature is also regulated by circulating water and ethylene glycol mixture, and I believe it can control a wide range of temperature, but in my experiment, I mostly use 20 degree as that is comparable to the room temperature. And yeah, the droplet size. So as the droplet shrinking, typically when it's small, as small as 5 micrometer in radius, that's our lower limitation for this technique. And then I would like to talk about the size and phase determination in our EDB measurements. So once we trap the particle, depending on its size and its composition and its phase, we collect, as you can see here, this is on the x-axis is the scattering angle centered at 45 degree and plus minus 15 degree on the x-axis, and the y-axis is the light intensity. And according from our data and compared to a mean theory fit, we can characterize typically we characterize 3 phases using such technique, including homogeneous, semi-dissolved, or spherical with inclusion, and also non-spherical inhomogeneous. You can see their phase, so we call this as a phase function. You can see the phase function are quite distinctive from these 3 status. And for the first 2 phase, we are able to determine the radius of the droplet using a geometric optics approximation method, and we are able to get a profile like this. And for the non-spherical inhomogeneous particles that look like this, it's we are not able to determine the size anymore in this case. So, and then I would like to point out, as the droplet shrink, the refractive index of the droplet is also changing. So in such technique, we also considering the changing in refractive index to, correct the droplet size. So typically the way we do it is to generate, a range of, make a range of solutions containing different concentrations and measure its density, refractive index of the solutions, and using a range of mixing rules in order to confirm its density parameterization, and also using the mole fraction mixing rule to confirm the changing in the refractive index during evaporation in order to make the size correct for our specific solution. And in terms of so we would also like to see how does this droplet look like. Here I introduce this technique called falling droplet column, which is also designed in Bristol. We use the exactly same dispenser to generate same solutions. And we collect these particles under the same conditions, for example, the same humidity and temperature as those in the EDB measurement. And we collect these dried particles, typically dried, onto this glass cover slip, and then we use it to do SEM images, do some post analysis. And here I would like to introduce the CLABS. So CLABS is controlled electrodynamic levitation and extraction of bioaerosols onto a substrate. So these are my colleagues who work in the virology and microbiology sector. They generate a bunch of aerosol droplets that containing real viruses. through the exact same droplet generators. So we all use the same micro dispensers, and they trap the droplet, in the also in a controlled humidity and temperature conditions. And depending on what we want to see, we collect these particles onto a substrate and to, for example, infect cells in order to determine the infectivity of these droplets. So it's a next generation virology technique, and that measuring airborne viral decay. And we are able to measure the extremely fast decay dynamics, and it's controlled both in gas phase and aerosol composition. For example, we control the CO2 gas phase. And then when we couple this technique along with the CKEDB measurement, we are able to, explore the decay mechanisms of these droplets. And I believe this is a video. You can see here these droplets are suspended in the chamber. And using both techniques, we are able to explore questions such as why does a virus decay in the air? And previous studies have explored the equilibrium state. So for example, in this very famous paper, where they use a slightly different EDB technique, where they trap two particles, but the humidity are changing during the measurement. So for example, it's a stepwise changing, so it's an equilibrium state. However, in the aim of this study is to explore the fast drying kinetics of water evaporation from surrogate respiratory aerosol. So our droplet is actually from humid and to dry conditions, for example, and then we want to examine the impact of mucin on equilibration of these droplets. And thirdly, we would like to characterize the timescales of phase change in drying respiratory aerosols. We think these questions will be helpful to investigate why does some viruses infectivity decay in the aerosols. So typically, this shows the typical results from the EDB measurement. So as you can see here, it's a 0.1 mass fraction of solute sodium chloride, where we have it's equilibrate at higher humidity, and it forms a inhomogeneous non-spherical salt when the humidity is below, for example, 45% for salt under the room conditions. And also we were able to measure the artificial saliva, but without the organic part in mucin, because we particularly want to investigate that in the later phase. So here you can see that this is the artificial saliva drying at different humidities in this case, and you can see from the falling droplet column, we collected these images at these relative humidities. And then this is just to show the DMEM solution, the Dulbecco's modified Eagles medium solution, drying at different humidities. As you can see, yes, when the humidity is high, it equilibrates with the environment, and when it's drying, we observe such morphology of the DMEM droplets. And so here in this slides, I listed, how does the experimental data look like. So typically you see we have one probe to, infer the relative humidity of the gas phase, and we have a sample evaporation profile. And combined with this, from this kinetic measurements, the equilibrium, we are able to, it's possible to infer the equilibrium hygroscopicity response for different surrogate solutions from, EDB measurements. And here is to show the hygroscopicity profile of these surrogate solutions that I measured through EDB measurements. As you can see here, so on the x-axis is the water activity or the equilibrium relative humidity. On the y-axis is the diameter growth factor, which is, the wet diameter versus divided by dry diameter. And we can see here, so typically we compare it with salt, sodium chloride, which is a well-established data. And here we plotted MEM, so the other type of cell culture media that contains large proportion of sodium chloride and much less glucose compared to DMEM. And you can see its hygroscopic curve is very similar to salt. And then we have the DMEM, which is slightly less hygroscopic, probably because it contains more glucose and organic compounds. And then here I compared two formulations. One is the artificial saliva from Wu et al., which is, I think it represents the unstimulated saliva level, so which is more similar, compositionally similar to when we are resting. While, for example, we're eating, we have more salivas and there are typically more sodium bicarbonate will be generated and then more salt. You can see by changing its composition, we also altered the hygroscopicity of these solutions. And from the last graph, we can also convert the y-axis into the mass fraction of solute. So here you can see that we typically, when we make a solution, it's typically in this range because you're not able to make such a concentrated solution. However, when it's evaporated in the aerosol phase, we are able to get information subtract from such measurements. And yeah, it can be used to infer the equilibrium droplet composition at varying humidity levels. So by retrieving these hygroscopicity profile of surrogate solutions, we are able to predict the evaporation kinetics of any surrogate solution under any set of environmental conditions and for particles of any size. So here I would like to introduce the SADCAT model, single aerosol drying kinetics and trajectories model, which is free to use and open source on GitHub. And this is developed by my colleague Dan Hardy, where we use the mass fraction versus, for example, water activity and, the density parameterization as the input. And we can predict the evaporation of the droplet at, different conditions from the SADCAT model. And here you can see the blue curve is the simulation from SADCAT. So this is to simulate salt. So salt is a pretty basic one. And not only that, we also are able to compare the SADCAT model data that is very comparable to these complex mixtures such as, artificial saliva and DMEM that we measured in the experiment. And just to note, these are all normalized against the initial size. So next, I would like to talk about the phase behavior of these mucin-free droplets. So here you can see that. So the blue bars indicate homogeneous phase, and the dark bars is indicating the non-spherical inhomogeneous phase. So from the sodium chloride, and on the x-axis is a range of humidity. So typically we repeat the same humidity for 10 to 20 times, and then we can get such information of how many droplets are behaving like in which behavior. And you can see here when it's above, let's say 45 degree, we know that it's 45% RH is the efflorescence RH, so we know that the salt will remain liquid. And when it's below the efflorescence RH, we know it's gonna become inhomogeneous non-spherical particles. And similarly, we observe the same thing in the artificial saliva recipe that doesn't contain mucin because it's a mixture of salt. So when reaching equilibrium, so we also observe a different one, which is in the beginning the droplet is homogeneous. So these are subtract from our phase function measurements. So initially it will become homogeneous, and later it behave like this, which is quite complicated. But still we can size this droplet. So so this is a DMEM droplet drying at around 50% RH, and yeah, it's you can see it's complicated, and yeah, we observe such complicated inclusion or semi-dissolved phase, in these typically the environmental RH, I would say, between 50, 80, and so this is new for these cell culture media. So we don't really see that in the mixture of salt or pure salt. And then here we would like to add mucin to our discussion. So here we want to talk about the influence of mucin on droplet evaporation. So here is the droplet that contains mucin and also don't contain mucin, drying at 40% RH. So on the left-hand side is the artificial saliva formulation. On the right-hand side is the DMEM formulation. So we start from the left, we can see that so these are all like 10 or 20 droplets overlap with each other. So we can see that in the case of artificial saliva, when we add mucin to the droplet, it only slightly decreases the evaporation rate. However, so these points are when it's crystallized. The crystallization time is much more delayed when you add mucin. Similarly, we observe the same phenomenon in the DMEM droplets, where in this one we also changed the mucin concentration to these levels are corresponding to the health status of individual. For example, a smoker or infected by SARS-CoV-2, they might generate different level of mucin in their respiratory fluid. So here, by changing the mucin concentration, we didn't really observe any, significant impact on the evaporation rate, but it does, slightly, it's slightly slower than the non-mucin droplet. However, we still, observe a similar phenomenon that the crystallization time is delayed. And here is just an example of how does the phase function look like for the DMEM droplets that contains mucin and don't contain mucin. So on the left-hand side, it doesn't have any mucin. On the right-hand side, it contains 0.5% weight per volume mucin. So you can see on the left-hand side one, the homogeneous, the droplet remain liquid until this point, and it forms non-spherical particles as indicated in the red dots. However, when we add mucin to it, it exhibits a slightly different behavior. So it evaporates, and then it forms kind of a semi-dissolved inclusion phase, and then it's become non-spherical. Now we are not able to determine the size anymore. So in the next slide, I just showed the statistics of this time point between different solutions. So first of all, we would like to see crystals. So as I mentioned earlier, when we add mucin to our formulations, including artificial saliva and DMEM, we do observe a delay. So these are 100 droplets for each solution, and we observe a delay in the crystallization time. So as you can see, the x-axis is time and the y-axis is the droplet count. And similarly in the artificial saliva, we observe a much more delay in the crystallization time. And this is important because we would like to, on the right-hand side is the results from my colleague working with SARS-CoV-2 airborne decay. So these are droplet infectivity at 2 minutes. We can see on the x-axis is a range of relative humidity. And the y-axis is the infectivity. We can see that, when the droplet is not crystallized, so on the right part, the infectivity remains higher than these that are crystallized. So we think that the phase behavior are important for these droplets. And then what's more interesting is that in this in a complementary measurement where we measured the MEM droplet, so similar cell culture media, we dried the droplet to 40% RH, so we know it will be dry, and then we rehydrate the particles, and then we circulate between 75, 40, and then 75 again, and then 40. So you can see that the phase of the droplet it has a tendency to become homogeneous after a while. However, when you cycle the RH, so once the droplet become crystallized, it has a detrimental effect to the viruses. So even though we increase the RH again, and the droplet, the phase has recovered to, for example, homogeneous or semi-solid. However, the infectivity is not going back. So it's like an irreversible process for decay for these viruses. And here is a comparison between just the SEM images for the artificial saliva droplet that doesn't have mucin versus has mucin. You can see on the left-hand side, it's a much more irregular, kind of much more rough surface, and you can see salt crystals on the surface on the left hand side. On the right hand side, you can see it's a much more spherical and slightly smoother surface of this artificial saliva droplets at 40% IH. And then for the DMEM droplets, we observe a similar phenomenon. You can see on the left hand side, when it doesn't have mucin, it looks like this, where it's much more rough surface, while on the right hand side, when we do have 0.5% weight per volume mucin, it looks like this. And if you recall the graph where I showed the complex behavior when the DMEM droplet that contains mucin drying, here we also found that if you increase the mucin, if you add mucin to the droplet, it exhibits the phase of inclusions. So the droplets remain liquid in the beginning, and then it becomes semi-solid, spherical with inclusion, and then become crystals during the drying process. So here is a record of how did these droplets that has mucin compared, also DMEM on itself compare in terms of the time to become inclusion. So you can see when there is mucin, it's much more earlier. This event happens much more earlier. And why this is important and interesting, because in my collaborator study where we used the mouse hepatite viruses, that is an alternative surrogate to SARS-CoV-2 virus, where we this is aerosol infectivity after only 30 seconds in the DMEM droplets. So in the DMEM droplets, we also added different mucin concentrations, and what they have found is that if you increase the mucin concentration in the droplet, and this is when it's at 40% RH, I believe. When it's dried, the droplet that has more mucin will has a higher infectivity in a short 30 seconds. So from there, we think that the the left graph would explain that, because the when it does have mucin, it's the time to form inclusion is much more early and had at a higher water activity phase. So to summarize, so from my measurement and also my colleagues' measurement on SARS-CoV-2 and the mouse hepatitis virus, we think that water transportation is crucial to understand the phase behavior and the corresponding, viral survival in respiratory aerosol. And secondly, the phase separation of salt and organic materials was absorbed in dried particles, and the complex phase behavior in the environmentally relevant humidity, as you recall, it's between 50-80 range, is likely the reason for the increased variability in microbial viability in the aerosol phases. And then lastly, from our measurement, you can see that mucin delayed the water transport and promoted phase change to occur at a higher water activity during aerosol evaporation, and the timescales with timescales consistent with the timescale of the effect of mucin on viral infectivity loss, on the mouth hepatitis virus, for example. And yeah, with that, I would like to thank Aerosol AAR, the American Association for Aerosol Research, and AS&T for publishing the article. And also, I would like to thank APSRC Center for Doctoral Training in Aerosol Science for their support during my PhD. And here is the website for the aerosol science. If you're interested, you can check it out. It's a big center, and each year we have 20 aerosol science researchers going through trainings in aerosol science and doing aerosol related research. And now it has I think we are in the cohort 6. So I was in the cohort 2. So we already have over 100 PhD researchers in the field of aerosols already. And check it out if you're interested. And then I would like to point out that now the project I'm working on is called Biodetection Technologies Hub, and our website has just gone live. So this is a very new website, and I think this is a really good platform to to talk about this and share it with the wide aerosol scientist community. And yeah, we would like to, so this is a 5-year project working on the biodetection. So we want to contribute, ultimately, we would like to, working on these aerosol detection technologies and to improve, for example, public health, plant health, or understanding the climate science, so we are open to collaborations, and in this field, if you're interested, also check out our page. So as you recall this little graph, in this paper, we mainly investigated the water evaporation process. Some of you might be very interested in the CO2 evaporation process in these aerosols. And good news is only 3 days ago, our articles on this topic and titled Measurements of Surrogate Respiratory Sensor Droplet pH and Implications for Exhaled Respiratory Aerosol and Airborne Disease Transmission was accepted by the ACS Central Science Journal. And I believe it will coming soon. And I hope if you're interested in my research, stay tuned. So you might be very interested in this article too. And yeah, thank you for organizing this event. And thank you again. I would like to take any questions. Thank you.
Thank you so much, Jahan.
Thank you. Thank you.
An amazing and insightful session. This is now time for question and answer. If anyone would like to ask any question for Jehang, it's a great time to take them now. If you want to mute yourself and ask directly, I think that's also possible. And if you'd like to share in the question or in the chat box, that also works perfectly.
Yeah.
Okay, we have one hand. I think you can go ahead and ask.
Hi, I'm Jiang. Thank you for your presentation. It's very interesting.
Thank you.
And thank you for your hard work on this as well. It's very, very interesting on the evaporation part. I just have a clarification question. You showed a few experimental data, like in slide 24 or other slides. You mentioned about that when your RH is very low, like below 40%, the the R square over R0 will, like, there's no data anymore. Can you elaborate a little bit more? What does that mean? Does it mean that the technology are not able to capture the size because they are solid now? Or, yeah, sorry.
Yeah, good question. So, yeah, in my graph, so from the EDB technique, as you, if you can remember, when the droplet is, non-spherical, we are not able to determine the size anymore. So in the actual, in the EDB data, we didn't show anything, further. However, in the SATCAT model, it's possible to estimate the crystallization and also the crystallization time as well as the crystallization size as well. So actually, yeah, we are able to extend that line.
I see. Then I have a follow-up question. So, like in slide 26, you present the yellow dot with inclusions and then the red as non-spherical inhomogeneous. So my understanding from your measurements, you mainly can see the homogeneous one. So were you also able to actually using the optical, whatever tools you're using right now to get information about the inclusions at all? Or is also something else you have to see it from the SEM, or you have to extend the line from the model?
Yeah, so previously, so back in 2019, so there is an article which is also in my slides. So the where so my the postdocs in our lab has tested, thousands of droplets of different compositions and also different phase of these droplets, and I think we are able to determine the semi-solid phase. And also there is another one called core shell. I think we are also able to determine. However, I don't think in my case, so in the case of respiratory fluid droplets, I don't think I see, for example, core shell structure, that perfect core shell. But this such semi-solid, I believe so, because we are able to characterize the size from the droplet, and also combining the phase function data.
Okay, thank you. They just really want to clarify on how you guys infer the phase status and then the shape from your technology. Very good work. Thank you very much.
Thank you very much. Thank you for your questions. Carl.
Looks like there's another question from Carl. Carl, do you want to unmute yourself and ask a question?
Now you can hear me. Hello, I'm Carl. I'm physician from Berlin, and I'm also doing research about the human aerosols from the lower respiratory tract. I got a lot of input now today. Thank you. Very good, structured, and well presented presentation. Yeah, there are so many aspects that well, I didn't know, but that are very important. One of my most amazing well points is that all that you described passed within seconds, right? So that was
Yeah.
3 to 5, 6, 7 seconds,
Yeah.
and then you all had these effects. So in real life, well, 7 seconds are nearly nothing. Do you have implications for daily life situations, or how to interpret this short time period?
Good question. So actually, in terms of the respiratory aerosol dynamics, it's a really complex. It's a very complex questions, because not only we have the water evaporation, the phase change of these droplets. You can see phase changes happens in seconds, but also we have the for example, the pH change of the aerosols, which happens in minutes. That is on a different time scale actually. So combined with all of these effects, what I'm trying to say is, for example, recent research has shown that controlling the CO2 level in the indoor settings would largely contribute to if we have a really high concentration of CO2 in the indoor settings, that will increase the infection of, for example, people at a great rate. So it's important to control for example, the gas phase conditions in the indoor settings. I think that's my take. But there is a lot of questions to explore still, in this research you saw that the phase change happens in seconds, and then in my incoming article I will illustrate a different time scale and on a different problem, and
So multifactorial, multi-complex, and
yeah.
there's yet no model to describe it perfectly. And
Yeah,
it's one
yeah,
issue of this cascade getting infected and so on.
yeah, yeah.
Thanks. Yeah,
Thank you. Yeah, yeah. There are
thanks.
a lot to
Great.
consider. Yeah.
Thank
Thank you.
Thank you, Carl. There's a question in the chat I'll take, after which we'll go to Ali's question. This is a question from Jonathan. Jonathan says, nice talk. Do you have any thoughts on how these results might change for smaller aerosols where the Kelvin effects can become important?
Good question. So in the beginning of my talk, I clarified that all my droplets are, we investigate the size regime of the upper respiratory tract. So typically we investigate these large particles that might come in from vocalization. Thoughts on how these results might change for smaller aerosols. So for the smaller aerosols, I think there is a different question because first of all, smaller droplets, they are from, for example, different generation mechanisms and different from generating from different site. So typically, maybe you're referring to lung fluid, and lung fluid has a very different composition compared to the droplet I was studying. There are a lot of surfactants in the lung fluid particles. But yeah, I don't know if I answered your question, but yeah, I don't really have too much thoughts, I think, on the.
Okay. I think Ali wants to ask us a question. Do you want to unmute and ask directly? There you go.
Hello, Jihan, and thank you very much for your nice presentation. Actually, part of my question is similar. I can answer also to Carl. I generate recently some particle in the range size of the human exhale, and I consider two conditions. I have mucin and no mucin. And about the evaporation rate, my results are very similar with GChem. And so for several hours, we keep them aerosolized up to 3 hours, and the change of the mucin concentration had a similar trend with GChem. And about the infectivity, also again, when we have a mucin, we have a higher infectivity compared when we don't have it. But the point is that when I change the concentration, my trend was a bit different with you. Not different. It means that I could not find any special trend. So how you can explain, for example, I can imagine when we have the mucin or we don't have mucin, we can expect what, but changing the concentration could not make any, for example, clear sense for me. How you think or explain the effect of mucin to keep the infectivity of the virus? How you can explain this, interpret it? Thank you very much.
Your question is, how does mucin protect the virus?
Yes, or because I saw the similar trend when I have mucin or I don't have completely mucin and use normal buffer. But when I change the concentration of the mucin, my trend was, and I could not find any special trend similar to you. How you can interpret the effect of mucin for increasing the similar infection?
Uh, a good question, but I feel like because I don't directly work with the virus, so I'm, as you saw, I'm only, focusing on the chemical measurements. I'm guessing there is another question, which is how does your virus respond to your environment? So I don't know if we are using the same virus. Do they have the same, response to the environment, to the droplet? Are you using SARS-CoV-2 or?
Thank you very much. Yeah, I got the aspect you approach, your approach were different from what I expect. Thank you very much.
Thank you.
Okay, looks like we have one more question from Peng. Says, thanks for the nice presentation. I have a question regarding the measurement of aerosol particle concentration at the particle source. We use some artificial aerosol generators, e.g. atomizer. Do you know if there is a way to measure the particle concentration for different particles? As you mentioned that those large particles will shrink very quickly. Thanks.
Right. Yeah, I'm reading the question again.
That's
okay. There is a way to measure the particle concentration for I think probably yeah, for example, adding another like humidity chamber before your sampling. For example, make it really humid to keep the droplet size remain at high humidity. I don't know if that's helpful.
Hello. Yes.
Yeah.
Yes, Jianhan, thank you very much for your presentation. Maybe I didn't see clearly what my question is. Actually, my background is mainly working on the simulations of ventilation and aerosol dispersion in different indoor environments. But we also need the experiments, usually the room level measurements, to validate our safety simulations. So usually for this purpose, we usually use some artificial aerosol generators in the specific rooms. And then we measure that at different locations. We measure the aerosol concentration at the different locations in the room. But there is always a problem that we don't know exactly what our aerosol concentration or particle concentration at the source, because you know that for the large particles or at the as a general room conditions, these large particles will shrink very quickly. Then it will very be hard for us to have some very accurate source concentrations at the particle source. So usually what we do, we just use the relative or normalized particle concentration to validate our CV simulation. Since you, in your presentation, I saw you did a lot of aerosol concentration measurement from the human, generated by the human. So I'm wondering, do you have any ideas that we can get some ideas for measuring the particle concentration at the source?
So do you measure the particle concentrations at a distance or using for example, aerodynamic particle sizer? Or?
Yeah, we use some particle counters at the different locations to measure the particle concentration.
But you don't know before it's shrinking.
Yes.
I think it might be useful to look at the SATCAD model where you can input, if you know the solution you're making, so you know the compositions of your droplet, and you are able to, and then if you know for example, the ambient humidity and temperature level, you will be able to simulate a curve, the evaporation profile of your solution, and from there you know the size of the dried size, and also the initial size of the evaporation. That might be helpful, but I'm happy to discuss more if you want, and I can share my email in the chat again.
Yes, that would be nice. Thank you very much.
Thank you.
We have one more hand up. Julian, do you want to unmute yourself and ask?
Hello, Jan. Thank you for your
Hi.
presentation. I was just wondering, because you said pH change is a matter of minutes. However, you've shown us that the phase changes and the evaporation is more a matter of seconds for the sizes you looked at. I was wondering, because for me, pH only is a phenomenon of liquid state. So I was wondering if pH change is only interesting if you look at only the part where the aerosol is still liquid for the RH below the efflorescence RH, or the whole phase above the efflorescence RH, or does it concern all the RH values? I don't know if it's clear.
Yeah, nice. Very good question. Thank you. Yeah, that's a good question. I don't have an answer, but yeah, as I mentioned in my next study, I would like to point out it's all in liquid phase, because I don't know how to define the pH when it's crystallized. Yeah, it's a very good question, and hopefully we can figure it out in the future.
Okay, thank you.
Thank you.
All right, looks like we've come to the end of today's session. Would like to especially thank our guest speaker for sharing the results from our research with us at this AS&T lecture series. We'd also like to thank everyone for joining, registering, and attending this session. And I hope you enjoy the rest of your day. Thank you so much, everybody.